Having examined the monomeric form of nucleic acid, let's now consider the polymeric form. Polymerization of nucleotides produces nucleic acids, the monomers being joined by 3' to 5' phosphodiester bonds as illustrated below. This means that the 3' hydroxyl group of one nucleoside is joined to the 5' hydroxyl on the next. Note the presence of the phosphate group joining these residues, and that it is an ester group with a phosphorus rather than a carbon atom. Because the phosphate is esterified to two nucleosides, it is a phosphodiester bond. This structure forms the backbone of all nucleic acids. Nucleic acids are numbered from the 5' to the 3' end, since this is the order in which they are synthesized and the order in which the information within them is read. DNA, or deoxyribonucleic acid, is built from deoxyribonucleotides. We can represent the primary structure of DNA in two ways. First, we can simply list the order of bases by using single capital letters to represent the identity and sequence of the nucleotides. A more complete representation is shown below, with single letters representing the identity of the bases and vertical lines to show the positions of the sugar moieties attached to those bases. A diagonal line through the letter P represents the phosphodiester bond. Note that the 5' end contains a phosphate or P, and the 3' end the terminal hydroxyl. Another common notation uses the same kind of single-letter code, but also includes a lowercase p to indicate the position of the phosphate group. The letter P is placed in front of the capital letter to indicate a 5' phosphate, and is placed after to indicate a phosphate at the 3' position. To distinguish RNA from DNA, a lowercase d is placed in front of the capital letter to indicate a deoxyribonucleotide. The absence of the D identifies a ribonucleotide. These are just various ways in which we can simplify and shorten our notation of what are often very long and complex polymers.